for somebody. Hello, welcome everyone. We're starting the webinar today, our weekly update. This is Sabrina Paganoni, and today I'm so excited to be here with our fantastic guest speaker, Dr. Heiman-Patterson, who is a PI for our trial. So as always, we'll provide enrollment updates. But to start, I want to turn this over to Dr. Heiman-Patterson, who will present her site.
Thanks so much. Thank you, Sabrina. It's a privilege to be here and talk to people who are interested, pals, CALs, anyone who's interested in the platform trial. It's such an innovative trial. I mean, it really has made huge progress. We're going to complete in 4 years what might take 12 years, for instance, in terms of number of drugs you can run through. And I'm sure you all know that. But I'm going to tell you a little bit about our center. We're very privileged to participate. If I could have the next slide. Who's moving? Good. Great. Oh, there I am. Well, I know what I look like, so we can move along. You'll get used to my sense of humor. The MDA ALS Center of Hope is at Temple University. It's a big mouthful, the Lewis Katz School of Medicine, and it's really, supported by the collaboration between Temple, the Muscular Dystrophy Association, and the ALS Hope Foundation. And that's how we can do the breadth of things that we're able to do that are, really necessary to do our best. Next slide. We all know that ALS is a family disease. I don't need to tell any of you that this affects everybody in the family. And next slide. And in our center, our multidisciplinary center, which is the centerpiece of the entire Center of Hope, we feel that it takes a team approach, and that as the team, we are your extended family if you're a PAL in our center, that we have to provide the same support and comfort of a family that you need to be able to reach out to us at any time. And of course, this is my team. And along the side of the slide, you can see we have physical therapy, occupational therapy. We do assistive technology. And I'm going to spend a little bit of time talking about that. That's something that I also feel very strongly about, is leveraging technology. We have, of course, speech, language, nutrition, respiratory therapy, mental health, mental health, nursing, and a huge clinical research component, because after all, as you'll see, that clinical research is one of the pillars of our center. And at the center, of course, as all centers, many of you, I'm sure, who are PALs are going to multidisciplinary centers, and we do symptom management, as well as make sure that you're on the drugs that you tolerate that can modify the disease in the here and now, while we really look to the future and clinical research that pushes new drugs forward, but also pushes understanding of the disease, biomarkers, and tissue banking forward. Next slide. Next slide, please. Oh, good. So, if you look at our center, you can see how we have both the clinical piece and the basic lab piece. And in our multidisciplinary clinic, clearly the most important thing is taking good care of people, making a difference to folks living with ALS with clinical care and education. But we also feel strongly about clinical research. And with a little bit of time, I'm going to talk about our brain-computer interface, that leveraging technology. But we also have several other clinical research projects, including looking at differences between phenotypes of ALS, people who have looked different between each other, what we call predominantly upper, predominantly lower motor neuron, and looking at oxidative stress in the different phenotypes. And I'm eclectic. I like oddball things. So I'm very interested in the GI tract in ALS and autonomic involvement, because I'm a little bit, I figure it's an area nobody else is interested in, so I can pursue it. And we have found changes in the GI tract and respiratory care. And in terms of trials, we are so privileged to be part of the platform trial. I can't tell you how exciting this is to us as a center to be able to provide this. And we have been actively enrolling. We have a wait list we're working through as fast as possible. We didn't get up and running thanks to COVID until January. And over the last 8 months, we've been pushing and increasing our numbers per month as fast as we can. And we love this trial. We're in all 4 regimens. And of course, we're able to have less people in, and you know this, and I'm sure Sabrina, Dr. Paganoni is going to go over this. We share the groups share a placebo group, so you have more chance of being on drug. It's a very novel design, and it's really going to accelerate clinical research in terms of finding new drugs. And we've also have several other trials going on, the very exciting AMX0035, which is going into phase 3, the Phoenix trial. And we've done oral Radicava. We're doing some biomarker refine and a couple of other drugs, as you can see. And then in the lab, we're just want to stress that I think it's really important to collect tissue. What's the best way to learn about human disease? Human tissue. And while iPS cells are exciting, human tissue is really important. And then we have a large project that looks at, in the animal models of ALS, what genes modify disease progression. And finally, we use those animals also to test potential therapeutics. You can go ahead to the next slide. That was good. That was fine. Thank you. And so I mentioned this. So we have the genetic modifiers, and we have found an area on chromosome 17. We're in the process of writing up actually 2 or 3 papers now on those genetic modifiers and the commonalities in several different models of motor neuron disease, those shared areas of modification that really may give us insight into motor neuron degeneration. And as with everything, we stress collaboration, and we've established a consortium of groups that will work together to translate that information to people, to look at into human disease and whether or not those same modifiers are operant. And I already mentioned some of these others, our biomarker initiatives. Let's keep going because we're limited. The other thing that I wanted to spend a little bit of time on is something I really feel is important, and that's the brain-computer interface and really leveraging the technology. What really upsets me and is that we have technology in the here and now that can make life better for folks with ALS, and it's not being leveraged. And so we are really working on technology to enhance independence, hands-free and speech-free, not only for communication, but also for home control and even for power wheelchair we're working on. Next slide. So the brain-computer interfaces leverage the use of a brain potential called the P300. When I look at something that I'm interested in, be it a letter or an icon to control our environment, it sets up a potential called the P300, which causes a brain wave that can be detected with an EEG cap. And that brain wave is detected and translates into an action, like that person was looking at the letter A, or that person was looking at the light to turn on the light. And that's then translated into a command to do something. And this has been developed for years. And in the sort of upper right-hand picture, you see a fellow with an EEG cap looking at a computer screen with letters. And look at that system. I mean, first of all, his head has to be just right. The screen has to be just right. There's 2 computers needed, one to present the targets, the letters on the screen, and one to decode it and translate it into an action. And there's this electrode cap that's 32 electrodes. You got to put it on in gel. It takes 45 minutes, and there's all kinds of wires all over the place. Well, and it costs about $25,000 to put together. Well, that isn't going to be really pragmatic now, is it? So we have begun to look at home-based systems using that P300 that are functional, that can do environmental control and long-term run a wheelchair even, that are independently usable. That is, the person can use it without a caregiver turning it on and off, that they will be able to independently turn it on and off. And that it is affordable. And so we've started using off-the-shelf components. So here, what you can see in the lower right hand is a fellow with augmented reality glasses. That was my first idea. Put the screen on glasses. It's always in front of your face. It doesn't matter where your head is. It'd be easy to control. And you can see in the upper corner the icons for a home environment control, TV, and so forth. So we started working with that, but the problem was that, you know what, AR glasses weigh about 1.5 pounds, 2 pounds, pretty heavy for somebody with ALS to control or be comfortable with. Next slide. So we came up with the idea that let's get dollar store glasses, put a transparent LCD screen on those dollar store glasses, and that would put your icons right in front of you, no matter where you were. It doesn't weigh very much. Let's simplify that system and use one computer. And in fact, for that, we have been using a Raspberry Pi and a battery pack to keep that Raspberry Pi going. And it turns out that the cost of the LCD screen is $10. The cost of the glasses is $1. The cost of your single board computer and the battery pack is about $105. And there's a commercially available headset that's the electrodes, that's dry, has 8 electrodes, slips on the head, and costs $800. Under $1,000, I have a BCI that can control a home environmental control. You have to also buy that. Final slide is just showing you, unfortunately, our video isn't playing today, but that's okay. This is the glasses we created. That's the screen. Here's the cap, and this is hooked into our Raspberry Pi down in the lower left-hand side. I realize you can't see my pointer. And this video just showed how you could select, and it has about 80% accuracy right now. And we're trying to develop all Bluetooth connections, and we'll be beta testing it soon, hopefully in the next 6 months. So, and we have a similar system working for a wheelchair that we're working on. So this is my passion, obviously, bringing technology to folks along with clinical research. And because of time, I couldn't go into, I could have gone on for an hour, but Sabrina told me I could only talk for 7 or 8 minutes. So thank you very much. And I hope that this tells you a little bit about our center and our philosophy. And also you get a flavor for how exciting we find the platform trial. And I really appreciate this opportunity, Sabrina. Thank you so much.
Thank you. Thank you so much. First of all, we love working with your site. And I have to say, I mean, you have been such an inspiration and role model, literally for generations of researchers and physicians, including myself.
I try not to think about that. I am 70 next week.
Wow. You are really, I mean, a leader, and I mean, phenomenal. Thank you so much for coming and for really training so many of us, mentoring so many of us, and for all the work. Clearly, your passion and dedication really is clear from your presentation, and hopefully we can have you back very soon. The only reasons we keep these presentations short is because we use it for Q&A, and we see lots of questions for you. So let me just finish the presentation with our usual weekly updates on the platform trial. You know, we've been asked by the community to provide weekly updates, which you are really happy to do. And so actually, if you can click a few more times, we see the arrows. This is again a novel approach with this platform trial. Thanks to the work of so many sites like your site, we can test multiple drugs at the same time. Right now we're testing 4, and we presented this in previous weeks, but essentially the beauty of this approach is that we can accelerate the timelines of drug development, which is really what we want to do. And so moving on to the next slide, many of you are following us on a weekly basis, and you can see here in the numbers that we continue to make progress. And specifically, if we look at the bottom part of the slide, you can see that when looking at the numbers by regimen A, B, C, and D, all regimens are making great progress. The target number of individuals within each regimen is approximately 160. and we're basically getting there. In red, you see the number of people that are already randomized within each regimen, and in blue you can see our expectations based on the people that we are screening. As you know, there is a process for getting into the trial. So we first screen participants, and then hopefully everyone who is in screening will be able to be enrolled. And so again, we are really nearing the end of enrollment for the first 3 regimens, which is great. And then obviously we'll continue to work on regimen D and add regimen E shortly thereafter. So again, great progress. So thank you to all of you who are participating or supporting us in many ways. Really, this trial will not be possible without your partnership. And so one of the things we love to do the most is really to connect with the community. And if you can go to the next slide, we have several opportunities for that. As always, today we're joined by our patient navigation team, Catherine and Alison, who are on the line. Thank you so much for joining. Please contact them anytime. They have phone number, email. There's a series of QR codes on these slides that you can find to really connect to our online resources directly. And if you go to the next and last slide, we can see a list of upcoming guest speakers. So next week, rather than meeting with a site, we will meet with Jennifer DiMartino, who is the executive director of ALS1, one patient foundation that has done really a lot of work and to support the trial and several other research projects. So we're very grateful to them. We will skip the webinar the following week because of the holiday, and then we will meet with Dr. Goyal from California. So we're really traveling around the country this summer to meet all our wonderful site PIs. And then we will skip a week in October because that's the week of our annual scientific meeting. And so again, we look forward to continuing to providing updates to all of you, and hopefully we'll hear about completion of enrollment very soon. You'll be the first to know on this webinar. So with that, I'm going to ask Alison to help us moderate the Q&A. Again, we use most of the time for Q&As, and actually it's great that you are on, Dr. Heiman-Patterson, so we can ask you a few questions. I see lots of questions in the chat for you specifically. So Alison.
Thank you. Yes, we've had a lot of questions come in, but please feel free to keep adding them into the Q&A as you think of them. Dr. Heiman-Patterson, first question for you is, what are you testing on PALS to see if the microbiome is not healthy, and how are you helping your patients in this area?
Okay, so I'm not doing microbiome work specifically. I'm actually looking at GI motility. It turns out that, you know, a lot of folks have GI symptoms, constipation, abdominal pain. And I got to thinking about this when I was, well, over the last 10 years. And it turns out that the autonomic nervous system is probably involved in ALS. And that's the system that regulates your blood pressure, your heart rate, and also your GI tract. And so we originally looked at the GI transit time, and it turns out it's slow. I then said to myself, why is that? Is that because everybody's on anticholinergics, those medicines to dry them up that also give you constipation? Or is there something else going on? So I began to look at the GI tract itself from, thank God we have a tissue bank and we have autopsies. And so I took the gut during autopsies and started looking at the gut. And in fact, it's actually a rough draft is already ready. There is a decrease in the number of neurons in the gut, what we call the enteric plexus, that moves the gut along. And so we're pursuing that to see if we can then improve motility using some pro-motility drugs, looking at before and after treatment for the GI motility with what we call a hydrogen breath test. So I don't do microbiome, but microbiome is very important. And certainly there's been work done in the animal model, in the mouse model of ALS on the microbiome that shows that if you change it, the disease does better. There's been some conflicting results in people with ALS. And so that has yet to be pursued adequately, or we don't have the final, I shouldn't say that, I should say we don't have final results yet in terms of what is the microbiome and how could we change it. Should you do yogurt a day? I don't know, but it may turn out to be something like that. We know that there's a brain-gut interaction for inflammation, and inflammation drives disease. And so certainly it is not crazy to think that the microbiome or the GI tract may have something to do with propagation, with the progression of the illness. I hope that does something to answer your question.
Fantastic answer. Thank you.
Been up about 5 minutes of time, Sabrina.
No, so appreciated. And further to that, there's a question. Do you think ALS can be triggered based on the environment?
Okay, that's another long answer. We're all born with a genetic endowment. Okay, that's why, heart disease runs in families, high blood pressure, diabetes. We have a genetic endowment that determines our susceptibilities. And in people who develop ALS, it's my belief that it's their motor neuron pool that's got the most susceptible population of cells. And in the proper, with age, duration of life, age, and environmental exposures of certain types, depending on your genetic background, it may trigger the disease. I can't tell you that, lead is the culprit or any one particular environmental factor, but certainly there could be a role for environment and that interaction between environment and genome.
Perfect, thank you.
You can jump in at any time, comments, because I don't know, I'm not the be all and end all, I don't know everything.
Well, you might know the answer to this question, Dr. Heiman-Patterson.
Is it my middle name? I know my middle name.
No, I don't. There's a comment that says this has been a great presentation, so thank you. When would the glasses be available?
Well, we're going to be beta testing, and it's interesting, and I'm trying to decide how to make this freely available. You certainly, if somebody's interested in beta testing, when we get to the beta test, no problem. I'm trying to decide how to create a nonprofit that will make these systems for folks. These are all off the shelf. You can buy this and put it together, but you need the software. And then the really critical issue to me is when something goes wrong, who are you going to call? Ghostbusters? I don't know. Who are you going to call? So we need to have an infrastructure of people who can help house with the technology to keep things running when they get them in their homes. So we're coming up with some ideas on how that can happen in a nonprofit way, where we're just trying to get the cost of the infrastructure. It's going to take a biomedical engineer, a software engineer, to do this, because when you have a problem with the system, but the system itself will be able to be putting together, I would think, the prototype by the end of the fall. We're going to be looking at it, and people, and if you want to test, just contact me, and we'll try to, if you're within a reasonable distance, we're going out to the house. If you want to travel, we'll try it. No, we love that. And the wheelchair, I hope, will be ready by the spring. And that we're using LiDAR, so you don't bump into walls, and we're trying to figure out how to GPS your home so you can just tell the computer, tell your glasses where your chair should take you. So that's going to be a little bit bigger nut to crack, but we have a prototype already for the eyeglasses and environmental control.
That's amazing. So about the HEALEY trial, what excites you the most about this trial?
Well, the drugs themselves are great drugs. They all have good targets. I mean, and that's one of our problems, is there's so many targets, we can't try enough drugs at once. But this trial enables us to look at several different types of targets, buttressing the mitochondria, and energetics, inflammation, which is a target that's very, we all know that inflammation's involved. The problem is it's involved at several levels, and sometimes it's good and sometimes it's bad for the brain. And, and so getting a better handle on drugs that modify inflammation. And I can do that at the same time. I don't have to do one drug and then another. And I alluded to the fact that, if you took 10 drugs and you wanted to try them one after the other, it could take up to 10 years, 14 years. If I do them in this platform, you're going to be done in 4 years. You'll know 10 drugs. And we got to get through these candidates. There are so many more candidates. The other exciting thing is it shows how interested pharma now is in helping with ALS. We have more drugs, and poor Sabrina has to get these drugs on board, and Dr. Sikovic have to work on getting these drugs on board and choose the right ones. But there's more involvement, more companies interested in getting their drugs in the mix. And let me tell you, I alluded to I'm 70. I realized I'm doing this for 40 years now. Okay. Isn't it time to retire? What do you think, Sabrina? Should I retire?
You should not.
I'm going to go do stand up somewhere. So, 40 years I've been working on this. When I started, there wasn't a single trial available. I mean, they were trying cough medicine, dextromethorphan, and which we now know does work, but you got to get it into the central nervous system. And that's Nudexa. But we were trying things like dextromethorphan on people, and there was nothing. And then little by little we had myotrophin, and then we had CNTF, and the neurotrophic factors had their heyday of interest. But in the last 5 years, there's been an explosion, maybe 10 of trials, and now with the platform trials, one of our problems with enrollment is there's so many trials, people are spread out over several trials. That was never a problem before. That's a good problem to have in a sense, because it means we have more opportunity out there for folks. And this platform trial gives so much opportunity to look at several different drugs in a rapid way. And we're going to get information about biomarkers, which I didn't have time to talk about, but another passion of mine is the importance of biomarkers in tissue banking. I don't know, did that answer your question?
Yes, it sure did. And I'm going to keep going with you. I have a couple more questions. I want to make sure that we get those answered. And I do have a few for Dr. Paganoni as well. But Dr. Heiman-Patterson, do you feel as though a fecal transplant is worth exploring?
That fecal transplant. There are people, we shouldn't laugh, but yes, once we understand the microbiome. I mean, fecal transplants are used for some GI diseases already. And there has been some interest in fecal transplant in ALS, but we have to do it in the right way and with a cogent logic to it, and really understand more about the microbiome in ALS and how fecal transplants would work. Theoretically, it would modify neural inflammation through that brain-gut interface.
There's a comment to thank you for your passion and dedication to ALS, and I'm going to second that as well. Thank you for all of that.
Well, I don't know how you tell in a 10-minute exposure.
We just know. Dr. Paganoni, I'm going to jump to you for a couple of general questions. There's a question, does the platform trial have an independent data monitoring committee that would stop if the drug is not effective?
Absolutely, there is one, and that's one of the great features of the trial, that we really don't want to continue unnecessarily if a drug is found to lack efficacy. So we do routine checks for that. So far, all regimens have passed all of them. So again, that's a great feature of the trial.
Wonderful. If a regimen is found to be effective, can everyone in the platform trial receive that drug?
So we discussed that if a drug is approved by the FDA, that would become standard of care and would be allowed in the trial. Just like today, we allow Riluzole, Nudexa, or Radicava. So if a drug was found to be effective, again, as we discussed in previous webinars, we really planned the trial so that if positive, the results could support the submission to the FDA for a new drug. So we really hope to be in that situation where we have robust results. And if so, the trial has been designed with the statistical features that would allow, hopefully, the drug to move forward. And so again, we need the robust positive results first. But if that happens, then when the drug is approved, it will be allowed.
Wonderful. Do you have to be ineligible for the platform trial in order to enroll in an expanded access program?
Yes, so an expanded access program by definition is a program to allow people who are not eligible for clinical trials to get access to the investigational product while the product is being tested in a formal trial. So we do have an EAP companion or expanded access companion to the platform trial that allows access to a small number of people to access to these drugs. But again, they cannot be eligible for the platform trial. And I would say it's small for now because of several factors, specifically funding and drug availability. But as discussed previously, it's our intention to continue to grow this program. We just started, and we will continue to grow it to allow more people to have access. But again, the program cannot interfere with the formal clinical trial.
Thank you. Since each drug in the HEALEY platform trial targets different areas, are there any clinical trial participants taking all 4 drugs at once?
You can go, Terry.
No. We don't know how these drugs are going to interact. This is actually Sabrina's, but as you know, the PI of the study, but we don't know how these drugs are going to interact. There's no way somebody should be on all 4 drugs at once. I do believe that if you finish one drug, you can re-randomize into another arm or go into the open label. But it would not, our job is not to kill you, okay? And I know how many of you feel that you want every opportunity to take every drug that you can possibly get your hands on, but really, we need, as an ethical tenet of research is risk benefit and keeping you folks safe, and using 4 drugs that are in trial where we don't know all the effects yet would be not a good thing.
Yeah, and most of all, we also want to tell if each of them is effective, and the only way to do it is to test them one by one. Now, in a platform approach, should one be found to be positive, then again it would be allowed in the trial, and so essentially then they will be combined, but again only after finishing the testing, so that we can only allow combinations of things that we know are effective and then build on it.
Great, thank you both for that. Question about open label extension. Sabrina, Dr. Paganoni, can you please explain how the length of time for the placebo-controlled portion and the open label extension?
Absolutely. So when we opened the trial, we wanted to guarantee that people who choose to go in the open label extension can remain on open label extension until we know the final results of the trial. So in other words, now we project that people will be able to continue in the open label extension until we know the results of the trial. In other words, if at the end we know that the drug is not effective, then there would be no point in continuing the open label extension. On the other hand, if we know that the drug is effective, then we will do everything possible to continue those people on open label extension for much longer. So again, when you write the protocol, you kind of have to say, for how long you're gonna continue. But then again, the idea is if the results are positive, to continue to expand on that and prolong participation.
Thank you. I know we're out of time. I'm going to go. We have 2 final questions. Dr. Heiman-Patterson, do you help your patients with breathing techniques that balance the automatic system? And if so, how do you do that?
The autonomic nervous system? No, that what we do, we want to, the problems in ALS with breathing tend not to be the autonomic nervous system and triggering, but with muscle weakness. So initially we start out with breath stacking and expiratory muscle training. And in fact, one of the studies we're doing is looking at the effect of expiratory muscle training on PFTs and on the breathing tests. And then we follow very closely. And if your breathing muscles weaken, we may implement ways to keep your lungs clear, which we call pulmonary toilet, cleaning the lungs out. And that entails nebulizer treatments, vest, which is a way to percuss or pound on your back, only it uses a vest. and something to help you cough called the cougholator, and a suction machine. And of course, non-invasive ventilation, which I feel very strongly about. I think Sabrina knows that because it was some of my original work was in non-invasive ventilation. And we know that it clearly improves quality of life if implemented properly. It will make you feel better, be less fatigued, and actually works better than any drug we have to prolong survival or prolong life in a higher quality way. It also helps cognition, by the way, for those of you who want to be sharp. I'm going to go use some BiPAP when I go home, so I can improve my cognition. But you know, so we don't so much look at the autonomic nervous system in terms of breathing regulation. It hasn't been shown to be effective, but heart rate has, and blood pressure. And one of the things that I've run into, sorry, I'm running over here a little bit, but one of the things I've run into that has been really bothersome to patients is fluctuating blood pressure, where they go all the way up, and then they go, if you use a blood pressure pill, it goes all the way down. And this can be very annoying, very bothersome to folks. And it seems to, I think it really occurs later in the course of illness, and has occurred where I've seen it in its worst manifestation. It's been in people who are on ventilators for a long period of time. But that dysautonomia is what we call that, when the autonomic nervous system isn't regulated properly. And in fact, it was a patient who I really loved, who was on a ventilator and got that first got me interested in dysautonomias in ALS. That's what, you know, we all have personal experiences, and that's the experience that sort of pointed me to the autonomic nervous system. Who would think, because it's only supposed to be motor, but motor is the tip of the iceberg.
Thank you. And final question today for you, Dr. Paganoni. Is there any collaboration with other countries, such as with the UK and the MND SMART study?
Yes, first of all, I want to say that I'm so happy that there's other investigators, other colleagues who are working on platform trials and find this approach interesting. So there is one platform trial that was started in the UK called MND SMART. They have a different design, different inclusion-exclusion criteria. Also, they are using repurposed drugs. So essentially drugs that are approved for other indications, they are repurposed for ALS. Instead, we have drugs that have never been approved for any indication. And so I think it's great to really connect with different researchers and talk about the best designs and what we can learn from each other. Again, I think it's we have regular conferences, both for the sort of national consortium of people who work in ALS in the US, but also we have the annual international conference, and we always talk about, again, differences in design and what we can learn from each other. So I think it's really great that there's different groups that are looking at this approach from a different angle, and I think there's going to be a lot that we can learn and really contribute to the field as a community. Was that the last question, Alison? Or
that is the last question. So I just want to say happy birthday to Dr. Heiman-Patterson, and I'll turn it back over to you.
Not yet. I still have a week.
But thank you so much for again. I think if I'm correct, you opened one of the very first ALS centers in the country or in the world. So thank you for all of that, and really for being such a pioneer, a mentor, really an inspiring figure in the field, and for all your work on the platform trial.
Listen, it's all because of the inspiration I get from the courage of people living with ALS. You know, we need to make a difference. We need to do something. I just, yeah, don't get me started. I can't do enough. I just feel like my work is never done.
Thank you.
It's really, I didn't do it. It's the inspiration is people living with ALS, and that's why we all do what we do. So thank you, Sabrina.
Thank you.
But I don't deserve the credit. PALS deserve the credit.
Thank you. I think we should, that's a great way to end this. And thank you for all the audience and all your support. Thank you so much. Bye.
Thank you.